Hey everyone, Dr. D here, and in this video we are covering Exam 2 from our Hoefnagels Biology Concepts and Investigations textbook. This exam is going to cover chapters 4, 5, and 6, so let's go ahead and get started.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Explain stuff.
All right, let's begin this exam review. As always, please study all of your lecture handouts, all the material we covered in those lecture videos. I'm going to try to remember the most important and meaty topics for this exam review, but it's really up to you to study all the material to do well. But I'm going to do my best to highlight some of the more important topics for the exam here. So let's go ahead with Chapter 4, The Energy of Life. You should definitely know the definition of energy. It is the ability to do work, also to move matter, right? So you should know the definition of energy. You should definitely know the difference between kinetic energy and potential energy. Remember that kinetic energy is the energy of motion and movement, whereas potential energy is stored energy that is available to do work. And remember, potential energy can be based on position or structure. Then, do you guys remember chemical energy? You should know that chemical energy is based on the energy inside of the bonds of the molecule. And you should definitely know this, okay? You should know that chemical energy is actually a form of potential energy because of its structure, the structure of that molecule. You should know the first law of thermodynamics for sure. You should remember that you should know for sure that energy can neither be created nor destroyed. You should know energy can be converted, energy can be transferred or transformed, energy uh, can be conserved, but energy cannot be created nor destroyed. You should definitely know that. In fact, you should realize that that's the first law of thermodynamics, that energy cannot be created nor destroyed. You should also know the second law of thermodynamics. Definitely know that the second law states that the disorder, also known as entropy, of the universe is always increasing. I definitely want to remember this fact. Next, I definitely want to know the difference between an endergonic reaction and an exergonic reaction. You should remember, you should know that an endergonic reaction is one where bonds form and energy is required, whereas an exergonic reaction is one where bonds break and energy is released. Try to remember those facts. Also, do you remember oxidation and reduction? Remember Leo says Gur, right? Loss of electron means oxidized, gain of electron means reduced. And I want you to remember for the exam that not only does oxidation mean loss of an electron, but that usually releases energy. And you should remember that reduction means the gain of an electron, and this is usually associated with requiring energy. Reduction requires energy, while oxidation releases energy. So keep those facts straight. You should know that ATP is the energy currency of the cell, and ATP is an example of chemical energy, which is also a type of potential energy. And do you all remember that I can, I often add clicker questions to the exam. So for instance, clicker questions may appear on the exam. So I would want to review all of the clicker questions. So for instance, which reaction is likely coupled with hydrolysis of ATP? DNA synthesis, yes. Converting proteins into amino acids, no, because that's breaking something down. And when you break things down, that releases ATP. or C, joining monosaccharides together? I would say E, A and C are correct. Next, you should definitely know what an enzyme is. You should know a few things about enzymes, right? You should know that an enzyme is a protein. It's usually a protein. An enzyme is a catalyst. Catalysts speed up reactions. And you should definitely know that enzymes do not get consumed by the reaction. That means that they can do the reaction over and over again without being consumed or destroyed. You should know that the substrate is the molecule that binds to the enzyme's active site. It's the molecule that gets broken down or acted on, and then the products are released. You should know one more very important thing about enzymes. You should know that the way that enzymes work is that they lower the activation energy. Remember this? The activation energy is the energy required to start a reaction. You should remember for the exam that enzymes lower the activation energy. And the more they lower the activation energy, the quicker reactions occur. You should know that cofactors help enzymes function. They assist enzyme function. Cofactors can be, vitamins. They could be metal ions like copper or zinc. Then you should know that inhibitors block enzyme function. You should definitely know that inhibitors block enzyme function. Non-competitive inhibitors bind to somewhere other than the active site and block enzyme function, whereas competitive inhibitors bind to the active site and block enzyme function. You should definitely know about inhibitors. And you should definitely know this one. You should know what negative feedback is. It's when the product of a reaction slows the production of more products. So you see in this metabolic pathway, A is converted to B by enzyme 1, B is converted to C by enzyme 2, and so on and so forth. Well, this final product feeds back to inhibit enzyme 1. You see, so that would be an example of negative feedback or feedback inhibition, it's called. You should know both of those terms, negative feedback and feedback inhibition. Next, you should definitely remember that temperature can affect enzyme activity. Salt concentration can affect enzyme activity. pH can affect enzyme activity. All three of these can affect enzyme activity. Next, you should definitely know that diffusion occurs down the concentration gradient. Next, you should know these terms: simple diffusion, osmosis, facilitated diffusion. All of these are important terms to know, as well as active transport and endocytosis and exocytosis. All of these terms are important to understand for the exam. You should know that simple diffusion is the passive transport of substance across a membrane, a biological membrane, and that substance goes directly across the biological membrane. And you should know that only small nonpolar molecules can cross the membrane directly by simple diffusion. So you should know that only small nonpolar molecules can diffuse across the biological membrane directly. Um, Polar molecules cannot cross by simple diffusion. Ions cannot cross by simple diffusion. Food particles cannot cross by simple diffusion. It's specifically small nonpolar molecules that can cross the membrane by simple diffusion. And by the way, you should know this. You should know that simple diffusion, osmosis, and facilitated diffusion, all three of these are examples of passive transport. Passive transport. That means that no energy is required for movement across the membrane, right? And the reason for that is because substances are diffusing down their concentration gradient. So All three of these are examples of passive transport, whereas active transport requires energy, endocytosis requires energy, and exocytosis also requires energy. You should know that nonpolar molecules, small nonpolar molecules such as oxygen or CO2 or methane, they don't require a transport protein to cross the membrane. If you do require a transport protein to cross the membrane, this is known as facilitated diffusion. Facilitated diffusion requires a membrane transport protein in order to allow the diffusion to occur across the membrane. So you should know these things about facilitated diffusion. They require a membrane transport protein. It's an example of passive transport. The substance is moving down its concentration gradient from high concentration to low. And those are the things to know about passive transport as it has to do with facilitated diffusion. And by the way, you would not need facilitated diffusion for small nonpolar molecules. You only need it for polar molecules or ions. Also, you should know that osmosis is the diffusion of water across a selectively permeable membrane. And you should remember that these tonicity problems are an example of osmosis problems where water is what's moving. So, for instance, when we place the blood cell in an isotonic solution, the water concentration on both sides of the membrane, inside and outside, are equal. So there's no net water flow into the cell nor out of the cell. The blood cell remains normal. But you should know that in a hypotonic solution, the water concentration outside of the cell is higher, which means the salt or solute concentration outside of the cell is lower. But recall that only the water is going to diffuse. So water diffuses from where water is more concentrated out in the solution to where water is less concentrated inside of the cell. So what do you need to know? You need to know that in a hypotonic solution or environment, water rushes inside the cell. Conversely, in a hypertonic solution, water rushes out of the cell. Definitely know what happens to a blood cell in a hypotonic, hypertonic, or isotonic solution. Which way does the water flow? That's what I need you to recall. And remember, during active transport, substances are moving across the membrane against the concentration gradient, and that requires energy such as ATP energy. And the sodium-potassium pump is an example of an active transporter. So clicker question number 5, why does the sodium-potassium pump require energy? Well, it's because it moves solutes against their concentration gradients. And do you remember the final concepts here? Endocytosis versus exocytosis. You should know that endocytosis is the bulk import of substances into the cell by formation of a vesicle, whereas exocytosis is the bulk secretion of substances out of a cell, also via vesicles. So here, let's do clicker question number 6. This solute is it polar or nonpolar? Well, it appears that the solute is crossing through a transport protein. And remember, only polar or ions have to cross through transport proteins. If these substances, if this solute was nonpolar, it could go directly through the membrane by simple diffusion. So it has to be polar. And does it require ATP? It does not require ATP because it looks like it's following diffusion. It's going down the concentration gradient. So A is the answer. And that leads to the end of chapter 4. Let's take a quick little break time with Gizmo and Wicket and be back strong for the next 2 chapters to continue our review. All right, welcome back from break time with Gizmo and Wicket. Let's start chapter 5 review now with photosynthesis. You should know that photosynthesizers are known as autotrophs or producers. You should know that all organisms, including eukaryotes such as plants and algae, are photosynthesizers. Specialized bacteria called cyanobacteria can be photosynthesizers, but no archaea, you should know this, no archaea have ever been shown to carry out photosynthesis at all. You should know that every ecosystem on Earth is based on photosynthesis. It's at the bottom of the food chain. Definitely know that fact. Next, you should definitely know the reactants and the products of photosynthesis. Remember this, you should know reactants mean what is required and products means what is produced, right? So you should know that the reactants of photosynthesis include CO2, water, and sunlight, and the products are glucose, C6H12O6 is glucose, and oxygen. Let's do clicker question number 1. The process of photosynthesis is evolutionarily advantageous for the organisms because it allows them to, remember this answer, it was make their own food. This was a tricky question because all of these answer choices are technically true, but the thing that's advantageous for them evolutionarily speaking is B. So to make their own food. Remember, they're autotrophs or self-feeders. Next, remember light. You should know, definitely know about light, that shorter wavelengths of light have higher energy than longer wavelengths. You should also definitely know that plants, plants and any kind of green leaf or tree or grass, the reason it's green is that it does not absorb green light. So you should know that green light is not absorbed by plants. You should know that pigments such as chlorophyll A absorb energy from light, and that is how we capture the energy from sunlight. And that leads us to clicker question number 2. Why are leaves green? So again, heterotrophs see green better? Nope. Plant pigments absorb green light? No. Remember, they do not absorb green light. Plant pigments absorb almost every wavelength except for green. That is the answer C. You should also know that on a leaf, a leaf from a plant, the underside of the leaf has specialized structures called stomata, which open and close to allow gas exchange. You should know that. And it's the mesophyll cells in the leaf that contain the chloroplasts for photosynthesis. You should know the structure of a chloroplast. Remember that stacks of individual stacks of thylakoids make up granum. And then you should know that the space inside of the thylakoids is called the thylakoid space. The fluid outside of these stacks is called the stroma. And there's on top of that, there's an inner and an outer membrane as well. Remember, that leads us to clicker question number 3. Do you remember clicker question number 3? Of the following list of plant structures, which is the second smallest? So the largest would be a mesophyll cell, followed by a chloroplast, which is an organelle, followed by the granum, which is a stack of thylakoids, and the thylakoids contain chlorophyll. So the answer is thylakoid. Now, you should definitely know that there are 2 main steps or stages to photosynthesis. Remember the light reactions, which happen inside of the thylakoid membrane, and the carbon reactions, also known as the Calvin cycle, which occurs outside of those pancake stacks, right, in the stroma. Okay. And you need to know the products of each. So let's talk about that. So this is what you definitely need to know. that during the light reactions on the left, the reactants are water and light, sunlight. And the three products of the light reactions, you definitely need to know the three products of the light reactions are ATP, NADPH, and oxygen. Those are the three products of the light reactions. And then let's look at the carbon reactions. Remember, the carbon reactions require CO2, okay? And they also require the ATP and the NADH from the light reactions, right? So technically, the carbon reactions require CO2 from the air, ATP and NADPH, and the products of the carbon reactions are sugar, like glucose sugar. And you should have, again, you should know that this image depicts the light reactions of photosynthesis. And you should know, again, that oxygen, NADPH, and ATP are formed. You should know that water and sunlight energy are required. And you should know that the way the ATP is formed is by this proton gradient. Do you guys remember? The electrons going on this journey, following the yellow lines, allows protons to be pumped, and those protons end up powering ATP synthase to produce ATP by chemiosmotic phosphorylation. So that leads us to clicker question number 4. How do the light reactions produce ATP? Select the one best answer. And again, remember, these clicker questions can appear on the exam, so I would learn them all. So A, potential energy stored in a hydrogen ion, that means proton, gradient is used to synthesize ATP. Well, isn't that it, right? Look at here. Look at here. During the light reactions, aren't we pumping protons and making a proton gradient? And that proton gradient is used to make the ATP. That's what it's getting at, right? So A is the answer. Potential energy stored in a hydrogen ion, which is another way of saying proton, gradient is used to synthesize the ATP. Next, you should know that the carbon reactions occur in the stroma, the fluid in the chloroplast, but outside of those granum stacks. What should you know about these carbon reactions? They are also known as the Calvin cycle. CO2 is taken out of thin air. You should know that CO2 is taken out of thin air and it is attached to an organic molecule. And you should know that this process of taking CO2 out of thin air is called carbon fixation. And the enzyme responsible for carbon fixation is called rubisco. So definitely know that rubisco is the enzyme that makes carbon fixation possible. And you should know what? You should know that these carbon reactions require CO2, they require ATP, and they require NADPH. And what do they produce? They produce sugar, like glucose. All right, let's check out clicker question number 5. How does a plant cell use ATP that it produces in the light reactions? Remember, the ATP from the light reactions travels over to the carbon reactions. So to fuel processes and reactions throughout the cell, no, that's for the ATP from the mitochondria. To fuel the carbon reactions, that's the answer. Yes, to fuel the carbon reactions. And awesome, that leads us to the end of chapter 5. We have one more chapter to do. Let's take another quick break time with Gizmo and Wicket, see what they're up to. Be back strong to finish off this exam review. All right, welcome back. Let's continue on with the final chapter for exam 2, respiration and fermentation, chapter 6. Now, recall that ATP is the energy currency of the cell, and it's through aerobic cellular respiration that All plants and animals and many microbes make ATP. Um, Remember that, yes, you should know that plants as well as animals undergo cellular respiration. You know why? Because plants also have mitochondria. Don't forget that plants have, in addition to having chloroplasts, they also have mitochondria. So plants can not only do photosynthesis, but they can also undergo cellular respiration. All right, and just like before with photosynthesis, you should also know the equation for cellular respiration. This is aerobic cellular respiration. You should know that the reactants are glucose and oxygen, and you should know that the products are CO2, water, and ATP. So this leads us to clicker question number 1. And remember, these clicker questions can appear on the exam. So do plants carry out cellular respiration? Remember, the answer is yes. So it has to be C or D. Yes, they require ATP like other living things, and respiration generates ATP. So yes, the answer is C. Remember, you should know the stages of cellular respiration, and you should know them in order. So you should know that glycolysis is the first stage, followed by the transition step, followed by the Krebs cycle, and lastly, the electron transport chain. You should know the ins and outs of each of these steps, right? So you should know, for instance, that with glycolysis, glucose is the reactant. You need glucose. And what's produced? Pyruvate, ATP, and 2 NADH. Again, during glycolysis, the input is glucose and the output is pyruvate, NADH, and ATP. And you should know the answer to clicker question number 2. What happens to glucose's carbon atoms during the process of aerobic respiration? Remember, glucose is C6H12O6. And during the process of aerobic respiration, those hydrogens are taken from glucose and handed to the electron carriers. Remember, NADH and FADH2. And so that means all that's left are the C's and the O's from glucose, and they are released as CO2. So the answer is during cellular respiration, glucose's carbon atoms are released as CO2. Here's something else you should definitely know. Glycolysis is the only one of the stages that occurs in the cytoplasm of the cells, or the cytosol of the cell. The rest of those stages occurs inside of the mitochondria. You should also remember the structure of a mitochondrion. Remember, there's the outer membrane, then you have the intermembrane space, then you have the inner membrane, and you should know that the matrix is inside of the inner membrane. Remember, the matrix is inside of the inner membrane. And let's solve clicker question number 3 together. Where is the mitochondrial matrix? Is it outside the outer membrane? No, that's where the cytoplasm is, or the cytosol is. Is it between the inner and outer membranes of the mitochondria? No, that's called the intermembrane compartment, or the intermembrane space. Is it inside the inner membrane? That's it. It's inside the inner membrane. Again, remember, glucose is required for glycolysis, and the 3 products of glycolysis are pyruvate, NADH, and ATP. And do you recall the answer to clicker question number 4? If 8 glucose molecules enter glycolysis, the net products will be how many pyruvate and how many ATP? Remember, it's 2 pyruvate and 2 ATP net for each glucose molecule. So the answer has to be 16 and 16. That's right, it has to be D. You should also recall that most of the ATP is produced after the electron transport chain by ATP synthase. Now, remember, we've covered glycolysis. Now, The next step is called the transition step, and you should know the ins and outs of the transition step. You should know that two pyruvates are entering the transition step by entering the mitochondria, and you should know the three products of the transition step. Pyruvate comes in, and what are the outputs? NADH, CO2, and acetyl-CoA, 2-acetyl-CoAs. So you should know those three products of the transition step. Next, you should know about the Krebs cycle. Again, you should know the inputs and outputs. So remember, the input is you have 2-acetyl-CoAs. These are the acetyl-CoAs. And the outputs are CO2, ATP, NADH, and FADH2. You should know those inputs and outputs of the Krebs cycle. And lastly, we covered the electron transport chain as well as this chemiosmotic phosphorylation with ATP synthase. What do I need you to know about this stage here? I need you to know that all of those NADH from the previous stages come here, and I need you to know that NADH drops off its electrons at complex 1, while FADH2 drops off its electrons at complex 2 of the electron transport chain. And you should know that the electrons travel through the electron transport chain where they are picked up by the terminal or final electron acceptor called oxygen. When oxygen picks up those electrons, it becomes reduced to water. So water is a product. Oxygen is required for aerobic cellular respiration because oxygen picks up the electrons at complex 4 of the electron transport chain. And do you recall that results in protons being pumped into the intermembrane space? Those protons chemiosmose. Remember, that's what the term chemiosmosis means. It means diffusion of protons. And chemiosmosis through this gradient is what drives your ATP synthase enzyme to make ATP via chemiosmotic phosphorylation. Isn't that neat? So those are the things I would want to know about the electron transport chain. And you should know that the electron transport chain is found in the inner membrane of the mitochondria. And so is ATP synthase is also found in the inner membrane of the mitochondria. And remember, the thing that makes this aerobic cellular respiration is that oxygen is the final electron acceptor, and it picks up the electrons at complex 4 right here. The only difference, you should know this for the exam, the only difference between aerobic cellular respiration and anaerobic cellular respiration is that in anaerobic cellular respiration, a molecule other than oxygen comes and picks up the electrons at the end of this particular ride, at the end of the electron transport chain. And you should know, let's see if you can beat Wicket on to this question. You should know the answer to this. Can humans undergo anaerobic cellular respiration? No, that's right, Wicket, who happens to be sleeping right over there right now. No, humans cannot undergo anaerobic cellular respiration because that would mean we could breathe something other than oxygen, right? So humans are not anaerobic cellular respirators. We are aerobic cellular respirators. And you should know that the only difference between anaerobic cellular respiration is that there's a different terminal electron acceptor than we have. You should also know that proteins, fats, as well as sugars are broken down by this respiration pathway. And then lastly, you should know about fermentation. Remember when fermentation occurs? Fermentation occurs when oxygen levels are low. So if we hit the gym too hard, we enter fermentation. If yeast cells are bottled up, they enter fermentation. And you should know that during fermentation, only glycolysis occurs. Only glycolysis occurs during fermentation. So only 2 ATP are produced during fermentation. And you should know that in humans, we produce lactic acid as our fermentation product, whereas in yeast cells, like the ones that make beer and wine, ethanol or drinking alcohol is the fermentation product there. And that leads us to clicker question number 5. What is the main advantage of fermentation over aerobic cellular respiration? And remember this one, it's that fermentation generates ATP even if oxygen is not present. That was the answer there. And then lastly, do you remember that photosynthesis and aerobic respiration are uh, converse reactions? One has a reaction going in one direction, and the other has the arrow going in the opposite direction. And this is why the gentleman was able to grow a plant inside of a closed off jar. And that leads us to the end of chapter 6. I hope you learned a lot. Again, I do have a video that goes much more in depth about cellular respiration. If you want to learn more details about that, I'll link it right above. I guess above my head here. You can take a look at that video where I go in a lot more detail about cellular respiration in case you need all those extra details. But I tried to touch on the most important facts that you need to know to do well on your exam. I wish you well, and I will catch you guys next time.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D.